When studying aerobic cellular respiration, we observed that oxygen was required at the end of the electron transport chain as the final electron acceptor. Thus, for organisms that are only capable of aerobic cellular respiration, oxygen is necessary. Such organisms are referred to as aerobes. On the other hand, organisms known as anaerobes are unable to tolerate oxygen, and in fact, it acts as a cellular poison for such cells. How is it that oxygen is necessary for some organisms but is toxic to others? Molecular oxygen and oxygen that is covalently bound in compounds are not poisonous. Oxygen becomes deadly when toxic forms develop. There are 4 toxic forms of oxygen: singlet oxygen, the superoxide radical, the peroxide anion, and the hydroxyl radical. Toxic forms are dangerous because they are strong oxidizing agents. In other words, they steal electrons. So why is that important? Well, what if they steal those electrons from important molecules in the cell, such as the DNA, or critical proteins or lipids? Such action may result in irreparable damage to the cell, thereby leading to death of the cell. Let's briefly examine the 4 forms of toxic oxygen. The first is the singlet oxygen. This form is an oxygen with electrons boosted to a higher energy state. This typically occurs during aerobic cellular respiration or can occur in photosynthetic organisms by the reaction of oxygen and light. This toxic form of oxygen is highly reactive. Pigments known as carotenoids can provide protection against the singlet oxygen by removing the excess energy. A second form of toxic oxygen is the superoxide radical, which can be formed from the incomplete reduction of oxygen at the end of the electron transport chain. Superoxide radicals are very toxic to cells, and thus cells utilizing oxygen produce enzymes to detoxify the radical. The enzymes are known as superoxide dismutases. These enzymes contain metal cofactors that combine with 2 superoxide radicals and 2 protons to yield hydrogen peroxide and oxygen. Anaerobic organisms do not contain these enzymes and therefore are susceptible to the irreparable damage that results from the oxidizing reactions that take place within the cell. Ultimately, it leads to the death of the cell. Hydrogen peroxide contains another form of toxic oxygen known as the peroxide anion. Aerobic organisms also synthesize enzymes to detoxify this toxic form of oxygen. There are 2 types of enzymes synthesized to detoxify peroxide anions. They include catalase and peroxidase enzymes. The catalase enzyme converts the peroxide anion into water and oxygen. One can easily detect whether a bacterial species produces catalase by applying a bacterial colony to a drop of hydrogen peroxide. This is known as the Catalase test. The formation of bubbles is indicative of a positive catalase test, meaning that the species produces the catalase enzyme. Peroxidase, on the other hand, utilizes a reduced electron carrier to break the peroxide anion down and yields water and an oxidized electron carrier. In the particular reaction shown, the reduced electron carrier used to reduce the peroxide anion to water is NADH. The 4th toxic form of oxygen is the hydroxyl radical. This is the most reactive form of the 4 and can result from the incomplete reduction of hydrogen peroxide. Due to the action of catalase and peroxidase, hydrogen peroxide does not accumulate in cells, and thus the threat of damage due to this toxic form of oxygen is eliminated. As we have seen, cells employ the use of carotenoids and the enzymes superoxide dismutase, catalase, and peroxidase to protect themselves against the toxic forms of oxygen. Another means of protection utilized by many cells are antioxidants, such as vitamin C and E, which provide electrons to reduce toxic forms of oxygen. Organisms can live at various concentrations of oxygen. However, be aware that if an organism is going to live in the presence of oxygen, it must produce the necessary enzymes to detoxify the toxic forms of oxygen. Strict aerobes can only grow in the presence of oxygen. They require oxygen as a final electron acceptor in their electron transport chains and do not run fermentation in its absence. These cells possess the necessary enzymes for detoxifying the various forms of toxic oxygen. Anaerobes obtain their necessary energy from either anaerobic cellular respiration or fermentation and do not have the enzymes necessary to detoxify toxic forms of oxygen. Because they lack the necessary enzymes, they die in the presence of oxygen. It is important as we look at the next term to break it apart. Let's look at facultative. Students new to microbiology like to link the term facultative to anaerobe and have a difficult time when they see it at a later point in the course associated with another term. According to the dictionary, the term facultative means occurring optionally in response to circumstances rather than by nature, or capable of but not restricted to a particular function or mode of life. With that out of the way, let's look at the term facultative anaerobe. If facultative means capable of but not restricted to, then these cells are capable of growth in anaerobic conditions but are not restricted to anaerobic environments. In fact, these types of cells actually prefer aerobic conditions and grow better in the presence of oxygen. In the presence of oxygen, such cells can utilize aerobic cellular respiration and in the absence of can carry out fermentation. Because fermentation has a lower energy yield per glucose molecule, growth is not going to be as abundant in the absence of oxygen as it will be when oxygen is present. Pay close attention to the image of the facultative anaerobe. Growth is shown to be its highest in regions of the test tube containing more oxygen and decreases as there is less oxygen. These organisms will have the necessary enzymes to detoxify toxic forms of oxygen. Aerotolerant anaerobes do not require oxygen for their metabolism, but are able to grow in the presence of oxygen because they synthesize the enzymes that allow them to detoxify toxic oxygen. Please notice in the image that their growth is not changed in the presence of oxygen. Again, this is because oxygen is not used in their metabolic processes. Our last group, the microaerophiles, are organisms that require oxygen for their metabolism, but are unable to tolerate the concentration of oxygen found in the atmosphere of 21%. These organisms have a limited ability to detoxify the toxic forms of oxygen and therefore can only tolerate smaller concentrations of oxygen. Consider how a microaerophile would grow in a tube like the ones shown in this image. So did you figure it out? If we were to observe microaerophiles growing in a test tube like that observed in the image, we would observe them growing slightly below the surface. They would not be able to tolerate the concentrations of oxygen present at the top of the solution. However, they would not be able to grow too deep in the solution either because they do require oxygen for their cellular metabolism. Nitrogen is another important element required for microbial growth. In fact, it is often a growth-limiting nutrient, meaning that if nitrogen is absent, anabolic reactions cease. This is due to the fact that nitrogen is required to build important molecules like the nucleic acids, proteins, and ATP, the energy currency of the cell. Cells can acquire nitrogen from organic or inorganic nutrients. Cells can also recycle nitrogen found in their amino acids and nucleotides. There are a limited number of organisms that can access atmospheric nitrogen, otherwise known as nitrogen gas. Approximately 79% of the atmosphere is nitrogen gas. Nitrogen gas can only be made available through a cellular process known as nitrogen fixation. Nitrogen fixation allows nitrogen gas to be converted to ammonia, which can then be used by cells. This process is vital to life on Earth because nitrogen-fixing bacteria make nitrogen available in a usable form to other organisms. More than 95% of the dry weight of a cell is composed of carbon, hydrogen, oxygen, and nitrogen. The remainder of the cell is composed of elements such as phosphorus, sulfur, manganese, copper, iron, and a few other elements. Phosphorus, like nitrogen, is a growth-limiting element. Phosphorus is an important component of organic molecules such as the phospholipids, nucleic acids, ATP, and some proteins. Sulfur is an important component of sulfur-containing amino acids. Sulfur groups can bind to one another to form disulfide bonds, which are critical in the tertiary structure of proteins and in vitamins. Other elements are referred to as trace elements because they are required in very small amounts. Some organisms are capable of synthesizing all the organic molecules needed for their cellular structures and metabolism from inorganic nutrients. Such organisms are lithotrophic photoautotrophs. Most organisms, however, must acquire small amounts of organic molecules that they themselves cannot synthesize. These molecules are referred to as growth factors. Growth factors include such molecules as vitamins and amino acids. Humans are an example of an organism that requires growth factors. We must acquire certain amino acids and vitamins in our diet because our cells do not have the biosynthetic pathways to synthesize them. Microorganisms that require growth factors are no different. Required growth factors must be acquired from their nutrition.